Okay, so we're continuing on. I don't know if I can pause this video, so we may have ended the other video and started part 2 now, or we might be continuing on and everything's fine. I'm not sure. We'll find out after the fact. So we were talking about behavioral isolation. Okay, so that's one thing. The next thing would be what we call habitat isolation. Remember, these are all ways that gene flow is prevented. In other words, it's ways that species are kept intact and ways that isolation between two species is preserved. Okay? So habitat isolation means, obviously, where you live. And so we can imagine that species might live in the same general region. So if we think about, let's see here, if we think about a map of North America, there's South America, North America. Okay, maybe there's a region here in the Great Lakes area, Great Lakes. So perhaps this is the region where this particular two species might live, but one of these species might live in, say, wetlands around the lakes, whereas another lives in open grasslands or deep forest. So because they don't share the same actual habitat, right? They don't actually exchange any genetic information. They don't have the opportunity to mate. Your textbook used the example of the northwest garter snake and the common garter snake. So it's called the garter snake because it looks like in the old days you'd use a garter to hold up your socks. So you had this little belt that you wore around your waist, and it had these little straps that came down, and the straps had a clip that would attach to your socks and hold your socks up on your legs. Right? We don't use them anymore. And these often had nice little colors and things. And so the garter snake has a striped pattern that makes it look like a garter belt, which we don't really use very often anymore. But the two species, the northwest garter snake, they both live in the same area. However, one prefers to live in meadows and basically dry areas. It never goes in the water. Whereas the other, the common garter snake, is more or less found near water. It's found in much more sort of sides of areas around lakes and marshes and wetlands. So it's a very wet environment that it lives in. And because of that, the two snakes then never really cross paths. And so that means their genes are isolated. So even though they're both two species that are very similar to each other, you can imagine if you went back in time, if you've got the western garter snake and the common garter snake, they're very similar. And when you look at them, it's hard to tell the difference. Perhaps they once were part of the same population that shared common gene pool. But some kind of environmental event separated the population. And so those that sort of moved over into the wet environment and those that were caught in this, say, dry environment, whatever this environmental event was, it would have separated the population. And these would have evolved in a different direction because now their gene pools are no longer mixing, right? The gene flow is no longer happening. And these ones would have evolved in a different way, to the point now where we have two completely different species, but they're still similar. So habitat isolation can lead to speciation, right? If you're isolated. And it also helps to keep species separate. All right. Now, the next one, or the next sort of kind of isolation, is what we would call a temporal isolation. And this has to do with time. The word temporal means time. Isolating, or isolation. Temporal isolation. And what this means, of course, is that species have a timing barrier between them. So they might have the same habitat, but they occupy that habitat, or they're active at different times. Let's put active at different times. They're active at different times. So, for examples, there may be two species of flower, for instance, but maybe the flowers come out at different times of the day, or in different seasons, or in different years. So you might have a field, and here's a flower that, remember, the flower is the sexual reproducing organ. So the flower appears when it's time to mate and exchange genes. This flower might come out in the early spring. And so during that time, the other species hasn't flowered at all. It's just like a little shrub or a plant. But then in the fall, this species has already flowered and it's over, and now this one produces a nice flower, and it flowers later in the season towards the fall. So that's a different time. That means that there cannot be very much gene flow between these two populations because they are separated in time. All right, so that's a temporal isolating mechanism. And the same could be true for animals. Two animals might be active at different times of the day, or they might mate at different times of the year. Many animals have mating seasons. Some animals mate in the spring, some mate in the fall. And so that would be activities that happen at different times, which isolates the ability of genes to flow. Then we have something called mechanical isolation. Now, the word mechanical usually means that you have to, it sort of has to do something with the way you work, your insides, the mechanics of them. And what this basically means is that they have incompatible, let's spell that right, incompatible body parts. Think about it. For sexual reproduction, if you have particular insects, you have organs for depositing sperm into the female, and they may be all different shapes and sizes. If you look at mammals, the shapes and sizes of a penis are very, very different. And so that's what we would call a mechanical means of isolation. It basically means that it prevents mating. It prevents mating because you don't have the right parts, essentially. Okay? So the genital anatomy is very distinctive, especially with insects. We find all kinds of strange variations in insect genitals. But even among mammals, we find that. You can kind of think about it's the lock and key theory, or lock and key method. If the key doesn't fit the lock, nothing's going to happen. Right? For plants, this also happens where you have variations in flower structure. Again, the flower has to receive the pollen from another flower, and that pollen has to land on the pistil. Remember that pistil, the female part of the flower, and at the bottom are the ovaries with the seeds inside waiting, and then you have the stamens that produce this is inside the flower, right? Well, when the pollen lands on the pistil, there are all kinds of interesting things that happen. The pollen has to sort of activate the opening of channels leading down through the style to get, and it has to break open and allow the sperm to swim down. Well, different shapes and sizes of pistils can interfere with different species pollen, keep it from pollinating the wrong flower. Okay? All right, so that would be that. And one more mechanism, and this is what we call gametic isolating, or gametic isolation. Now remember, a gamete, right, is the sperm and eggs. So this is basically how the egg is fertilized, how the egg cell is fertilized. There are different methods of fertilizing an egg cell. For instance, frogs, basically, here's a female frog, right? Sitting here like this. There's a female frog. Okay, it sits on the ground and it lays a bunch of eggs. Well, the male frog simply sits on top of the female, like this, and as the eggs come out of the female's body, the sperm is just deposited on top of the eggs outside. It's an external form of fertilization. Many fish also fertilize this way. So there's no penis, there's no inserting any body parts. Whereas in mammals, obviously, the fertilization is internal, inside the female reproductive tract, right? And so think about that. If you have the female reproductive tract, right, the uterus, and then the fallopian tubes and the ovaries here, right? Well, sperm has to make its way, has to be deposited here and make its way up the fallopian tube to meet the egg coming down. Now, different species have different biochemistry. And so a sperm from this species might be better suited to withstanding all of the female's defense mechanisms. Whereas a different sperm from a different species, if this was a cow and the sperm was from a pig, that sperm might not have the same ability to evade the cow defenses, and that sperm would be rapidly killed off by the cow's body before it had a chance to make it to the egg. So a gametic isolation is something that somehow prevents the eggs from being fertilized. Whether it's the type of fertilization that's different, or whether it's the body of the female sort of rejecting the sperm or killing or destroying the sperm before it can make it. Okay? In plants, the stigma, again, that stigma where there's the ovary, in plants, there are chemicals. When remember the pollen grain is a protein coat, and inside are the sperm cells. So when it lands on the stigma, the pollen coat has to either dissolve or break open or degrade to let the sperm out. And there are chemicals on the stigma that do this. So if the wrong pollen lands on the stigma, it won't happen. And the pollen will just basically be defunct. It won't actually open to release the sperm. Whereas if it's the right species, then this pollen will land on the stigma and there will be chemicals or enzymes that can break open the protein coat and release the sperm. So again, these are mechanisms that isolate particular species and essentially prevent gene flow from happening between two different species. And these are all things that happen before the sperm reaches the egg. Now, obviously then, we have also what are called post-zygotic mechanisms, isolation mechanisms. These are things that happen after fertilization, after the zygote has already formed. All right. And these have to do with, essentially, how the sperm and the egg sort of join together. And so, basically, remember the word hybrid, which stands for two different species being sort of put together, or two different races or breeds? Hybrid means putting two different things together. So we have what's called, this word occurs in all of these, hybrid inviability. To be viable means to be able to survive and to live, right? Remember the viable offspring that can reproduce and then reproduce again? Well, it's possible that you can get a zygote production. So you can have a sperm plus an egg that produces a zygote. And that zygote might even grow into a full individual, but that individual is sterile. It does not produce its own zygotes. Remember we said a species has to have viable offspring? This would be inviable offspring. So that can still happen. Even if the sperm and the egg do manage to get together, they produce an individual that is sterile. So the best example is the mule. A donkey and a horse are two different species, but they're similar enough that a zygote can be produced But when it does, it grows into an animal called a mule, and mules cannot have their own babies. So therefore, it breaks the chain, and that prevents the mule from passing on any genes or contributing to the gene flow. Okay? And of course, the tiger and the lion that I mentioned before would also count for that. The next is what we call hybrid sterility. Okay, so I realize now that I jumped the gun. And actually, the mule is a better example of this. Sometimes when I do these things, I keep them in my head, and I make little associations, and then I forget to undo those associations before I teach to you. So the hybrid inviability usually means the zygote itself is inviable. So it doesn't usually get to the point where it becomes an individual. I should have mentioned that. So let's cross this out. The zygote forms but dies before it grows up, before it basically reaches term in the pregnancy. It doesn't actually make it. So you're talking about a miscarriage here. So let's write the word here, miscarriage, to remind us of that. The zygote can form, but it doesn't quite make it to growing up. So it never becomes a fully fledged individual. It's this next one, sterility, where the zygote does form, and it does produce an individual. So sorry for the confusion, that's my fault. And the individual is sterile, right? Sterility. I should have known that from the words. So this would be the mule, the horse plus the donkey, and they produce a mule, but the mule cannot have babies. It is sterile. So in this case, you have a fully functioning individual, except for the fact that it can't have babies. Whereas here, the zygote is formed, and as it's developing inside the mother, something goes awry, and it's usually because there are genetic problems, right? The two sets of genes aren't compatible, and the mitosis process isn't working right. So mitosis begins to happen as the cells divide, and it doesn't work, and the zygote dies quite quickly. Whereas here, it can reach birth and become an individual. And then lastly, we have this thing called hybrid breakdown. And in this case, what you have here is sort of almost getting it right. So you have two species. They mate, and they produce an offspring. Now, this offspring is actually viable. It can then mate again and produce another offspring. But very quickly, the chain dies. These offspring are not viable. They live, but they cannot produce their own babies. So sometimes it takes a generation or two before the system breaks down. And so hybrid breakdown means that you can start off and you do get something in the first generation, right? So it would be like if a donkey and a horse produced a mule, and the mule had a baby, but then when those babies tried to have babies, it all fell apart a couple generations later. So in other words, the line cannot be preserved. You get excited because you're like, oh, look, we've got a cross. And then the cross happens, it has its own babies, but then eventually it fails. So this is a failure after generations. So let's put the hybrid breakdown or failure after a few generations. So it's almost going to work, but then it falls apart. Okay? All right. And so these are the mechanisms of speciation and the mechanisms that keep gene pools separate from each other. These are the mechanisms of isolation. All right? And so essentially, because they isolate, they basically are working against blending of the gene pool. So all of these are isolating mechanisms. That's important. And what they're doing is they are preventing gene flow. And when you prevent gene flow, you basically prevent two individuals or two populations from mixing their genes. And so you set them on two different evolutionary paths that diverge, right? And you produce new species. So, for instance, one of the stories I told you before, I'll just reiterate. In Africa, the East African Rift Valley, remember how I told you that there was a species that was ancestral to both humans and chimps. But there was a great massive ecological change when this basically earthquake opened up this giant rift, which separated the population, right? So this would be a kind of separating or isolating mechanism, and therefore the genetic drift or the genetic gene flow from here, from this side to the other side, was no longer happening. So what happens is the two populations begin to sort of evolve down different paths. This side evolves towards one thing, and this side evolves towards another. And again, if it happens over time and it happens enough, the differences will be so great that these individuals now are no longer compatible for mating, and they are no longer the same species. And this is what led to the differentiation between the chimpanzee and the human, right? It was an isolation. And so just understanding that isolation can be a geographical kind of thing or an event, but it can also be all these other mechanisms as well. Okay, we'll stop there. In our next video, we'll deal with types of speciation.